As with basically everything, we're going to start up with a quick intro to the workshop, presenting an overview and the things that are going to happen the next 2.5 days, not full days. And a few things like generally before we start, if there are folks here with hearing impairments or problems, we have the option, at least in some presentation, to turn on closed captions. So if you would like to have that, please let us know via the communication channels. And if you don't want to say it publicly, it's also fine. Just write us a private message, okay? For the folks who wish impairments, no matter what it would be, all the graphics except taken directly from publications should be high in contrast and color brand friendly. If you have problems there perceiving something, please let us know as well, so that we have the option to explain it better. To everyone, of course, basically continuing the point I was just making before, we will try our best to speak loud and clearly. If you have trouble understanding us, please ask. That's all fine. And again, if you feel uncomfortable doing that, just write us a private message here in the chat via Zoom, and that's all good. Continuing a few points for everyone. We hope you feel comfortable indicating a pronoun during live sessions. If you want to, please add them. You of course don't have to, but if you want to, please do. During the discussions and questions, it would be cool if you could say your own name so that we and all the others here can avoid mispronouncing it. And as for the content of the workshop, we try, of course, our best to present an objective overview, but being the people we are and the things we do, there's of course an inherent bias to such presentations in general, but specifically also here, based on our opinions and experience and the work we've done in the past. So we're looking forward to openly discuss everything we say here and comment with you all. And obviously, and I don't have the fear or thought that this was might not be possible, let's just give our best to create an open, supportive, and welcoming atmosphere for everyone during the next couple of hours and tomorrow as well. And we're actually gonna, oops, there we go. I don't know why it looks this, there we go, okay, yeah. And we're actually gonna start out with a bit of interactive question, icebreaker kind of thing, because virtual, we still get things going a bit somewhere else. And the question from us to you would be, do you already or did you already try to incorporate reproducibility into your own research workflow? And if you head over to the website, I just shared in the Zoom chat and enter that little number there, you should be able to get to our poll and provide a vote. Just for us to get a feeling what's going on in terms of reproducibility in your life so far.
Sorry to interrupt, Hera, but I presume you don't want the instructors to answer this, do you?
I mean, I guess for that to make sense, we would have to have color-coded responses so we know who's saying what. I mean, if instructors would put something other than yes, I think we would have to remove them from the list of instructors.
And to be honest, I think it's more interesting to see what the attendees distribution is. So while I was interrupting, do you encourage interruptions while we present in this format?
Oh, yeah, it's a good point. Thanks, Dave. Yes, obviously, interruptions, as long as it's constructive and questions are more than welcome. You don't have to wait the next 3 hours to ask a question like, where's the material? Okay, so just at any point in time, either raise your hand using the little Zoom thingy. Reactions always disappear when I share my screen, but you know there is this raise hand thing in Zoom. So either do that or just unmute yourself and with thoroughly, but maybe not screaming. Let us know that you're there and have a question. Okay, so I think we almost got everyone, 19 responses. Thank you all so much for taking part in that. And I gotta say, and I don't mean to sound like mean or rude, but I didn't expect that kind of vote. That's pretty cool. That's fantastic. If you already tried to, more than half of everyone that brought a vote tried or already is doing in reproducibility or working towards reproducibility, which is what we found. And only a few people didn't try so far. So with that, I actually hope that we can show you something new in the workshop. You've already been at it a bit. So I guess independent of if you've already tried to work with it or already worked towards reproducibility, you most likely have heard about this thing called the reproducibility crisis. That is not only happening in neuroimaging, but name the research field, it's happening there as well. It's a big common problem in science. And when you look around in the last couple of years and really putting in a year or a number on it, I think it might not be possible, but it's definitely around like the early 2010s that it became like really apparent and one more prominent that we have a problem regarding the things we do and the things we get out of there. And given that we want to understand something about the brain, obtaining reproducible results is something we should all aim for, basically. And this problem basically incorporates everything from certain bugs in software, certain methodological approaches people use sometimes, problems in education of people, or of course the incentive structures, the system itself. And the problem is, at least the way we perceive it, others as well, is that there's basically for the last couple of decades, there's been way too much trusting in science and not verifying. And this is of course something that should not happen. I mean, we should trust science. That's not the point. And we should be confident in the things we do and the results we get, things we read. But blindly trusting it is also not a good idea. We need to verify things. And the problem being very nicely summarized in this quote from Marvey 2015, saying that the broad adaptation of computers and experimental science has both enabled and obscured research. Because the problem is that if you run a certain code or analysis, you would usually assume that they run reproducible and stable. So if you rerun them a couple of times, different computers, different data, whatever, you would get the same results. But it's of course not the case at all. Actually, it's the contrary. And a lot of the things people use out there is actually not properly evaluated and tested regarding its behavior. And this, of course, becomes essentially especially problematic with the increasing sample sizes and convoluted complex models. We all know the buzzwords, big data, machine learning, deep learning, and we're all super interested in that. And it's not that these things are bad. That's not the point at all. They're very good. And I think, and we all think it's a good way forward. But adding more and more data and potentially with that noise to our main problem will just exteriorate, which is not good. And as I said before, really no matter where you look into, it doesn't have to be neuroimaging. Look at a study summarized here by healthcare from 67 target validation projects in oncology, only 21% could be reproduced. And as soon as it's about some sort of drug or a therapy or whatever, that's, if you don't care about reproducibility and basic research, okay, but you should definitely have it in clinical research. And again, this is across the board, also cancer research, economics, no matter what you look into. And this is a nice slide that I adapted from Felix Show Bro, because most of people in Germany might know. He's one of the groundbreakers over there, basically providing you a bit more detailed information across different research fields and the percentage of reproducible and unreproducible results. As you can see, there's quite a bit of variation in there, but overall, what you would like to have is, of course, 100% or close to 100% reproducibility, because otherwise, what would you do with the data if you know, or the results, if you know it's not reproducible? Do you trust it at all? Maybe, maybe not. So what is happening? And this is something we will try to address throughout the different sessions in this workshop. People, of course, make unintentional and unfortunately quite often also intentional mistakes. With the garden of forking paths, with all the different software we can use, all the different ways we can analyze certain data sets, the different tests we can apply. We have questionable research practices and p-hacking, unfortunately. There's also fraud and publication bias. And in this workshop, with the things we do, we cannot sufficiently tackle all of those, but quite a bit. And this is what we aim for also in this workshop. Because the idea is, or the main problem that we try to address right the different ways, in that everything is important. No matter what you do in your research workflow at any stage, this matters, and it has to be reproducible, because otherwise things along the way will get lost and become unreproducible. And I don't know about you, but quite often there's this dissociation between the ideas and hypothesis components of research project that you might have, that someone provides to you, and the results we would all see in a fancy presentation in journal. There are these two things. But what happens along the way is that researchers do a lot of things to get from one point to the other, using a large amount of tools and resources and steps to get there. And if we don't keep good track and make use of reproducible practices along the way of aspects, we get a problem. Because then going from this point to this point is basically impossible. Because in our opinion, the problem is, if we go in a rather classical research workflow, from project creation over data acquisition to analysis, results, and communication of results, if you would only share or make one part of these things reproducible, this would not be sufficient. What is sufficient is if we make every little thing reproducible and try to incorporate the respective practices. But this can only be sufficient if it's done in the right manner. So how can the respective steps of the research workflow be made more reproducible? And at this point, I give over to Dave. And Dave, you just let me know when I should advance the slides.
Yeah, thanks. So it's nice to just be able to add a little commentary to what Per has already introduced as many of the main missions that we try to undertake with Repronym. While he took the carrot approach, I'm going to take the stick just for the fun of it. And this is the so-called fear factor slide. And if you hit forward there, we will say that instead of talking about reproducibility too much, we should just do it, or else. There's a lot of things that are beginning to not happen if you don't consider reproducibility. You might not get funding. Different funding agencies are now putting into place more mandates that force reproducibility upon the folks spending their money. Annual funding may be held up if your current year isn't progressing reproducibly. More mandates for reproducibility and publishing are happening. So many journals are now beginning to enforce their data and software sharing policies. And if you can't get funding and you can't get published, you're not going to get a job. You're not going to get tenure. So this is bad. You got to do this. And most importantly, the late night comedians will begin to laugh at you if you don't behave reproducibly. And it's always annoying to be the butt of late night TV jokes. We're not going to watch that video clip, but that's in the slides, and it's a very telling and unfortunate clip. So if Yarrick were with us, he would be pushing the mantra of reproducible by design, embrace reproducibility from the get-go. Next slide, please. Per foreshadowed, that there's many steps that one can take in the reproducibility. He already showed that nice little workflow, and that has a study design, data collection, data processing, statistical analysis, and publication and sharing as part of that. Each of these can be broken into many steps. And while maybe what he used as none of those are sufficient until you do them all appropriately, the nuance I'd like to give to that is, since there's so many things to get completely right, I don't want people to not start because it's too daunting. Doing even little bits of that, even if the overall thing doesn't get completely there, each little bit that you do in your next study that improves your position on adding reproducibility is an important step. And together we will get closer and closer the more these technologies and techniques become mainstream with what you do. Next click. And so a graphical way, and this one more click, is to think about those steps on this sort of x-axis, and the next click is really the things that we want to do within each of those steps. So the commonalities to a lot of the Repronym technologies are best practices and activities that in each of the steps of the research process, we actively plan, we actively version control, we actually actively annotate and describe what we're doing, we actively use standards, and we actively archive and publish each of the stages through the process. And so you can imagine, most of the tools that you'll see that either Repronym is promoting or that exists, in the community, help you fill in, these steps along those practices. And so again, we encourage people to build up the tools and repertoires in this area. Okay, next slide. I just wanted to make sure there's a couple sort of just propaganda slides, just to make sure that we give credit to a lot of the people who are part of the Repronym effort. Again, you'll hear from a lot of them today and tomorrow, but it's a bigger effort than that, and a lot of initiatives that go behind it. The design around the teaching that we're doing today is around the core projects of data discovery, data description, and doing analytic processing sort of forms the three sort of core projects of Repronym. Okay, next slide. And yes, it's lovely that you're joining us today for 2 days, but Repronym is a lifetime. Anyone who's been touched in any way by us, we hope engages with us more continuously. So this slide just reminds you of our website, reminds us you of email and Twitter and monthly webinars and things like that. So I think I will stop there. I didn't remember that slide there. Okay. All right, so last thing I was gonna say, you got a a short introduction to the reproducibility problem, and it's nice to be able to do that quickly. We do this lecture on many occasions and sometimes in longer formats. So for the fun of it, in your slide set, I pointed you to at least 4 more versions of that same introduction that might be at a longer format. So just to peruse at your own leisure if you want to see the longer version of that. So thank you for letting me interrupt some commentary there, and I'll turn it back over to Per.
Yeah, thanks, Dave, for the overview and bring us through about Repronym. Basically, continuing from that, there's of course a little disclaimer again to the things we're going to show you and the things we're going to present. It's the things we worked on and Repronym has worked on the last couple of years. And it's of course only a subset of everything there is, heavily biased towards our experience and knowledge, and by no means complete. And we are aware that there's much more going out there, and I guess you are aware of this problem as well. It's just very hard to keep track of all the cool things that pop up. If you spend an hour on Twitter each day, there's just 10,000 new things you want to do and check out. So what's really important for us is that we would love to get your suggestions and additions and feedback so that we can further enriching our materials. So, but now, actually, for this workshop, our objectives, or basically the objectives for you, our objectives for you, is first of all, of course, gain skills. That is, know basics of reproducibility, focusing on neuroimaging, learn about tools and resources for reproducible neuroimaging analysis from us, but also others, and also how you can utilize them. With the idea being that we would like to empower you to start using these things for your own research right away. Of course, not 100% capacity, but start using it, and that then allows you to dive further into them in more detailed depth. Obviously, because we want to grow the community as well, people that do care about these things, it's important for us that you get involved. That is, you familiarize yourself with the reproducible neuroimaging ecosystem. That's not only us, but so much more, and the fantastic community behind it. And obviously sharing everything you've learned, which we hope will be a lot by the end of the workshop, to your home institution, to your lab. Share things widely. All of the materials, everything will be on the website or is already there, and it's open for you to reuse and share with the respective license.
I could just, for the fun of it, it's always easier to do these things if you're doing it with a team of other like-minded people. So that's why it's great to bring these things back to your labs and encourage other people to work on this with you, so that it's just easier with other local friendly people.
Yeah, especially considering that you will have a very, very hard time if you are the only one in your lab or research institute that does care about these things and uses these tools and resources. I did that for 3 years in my PhD, and it was not a good time. And it was a very happy once I was somewhere else with people that also did care about it. Your specific role within this workshop. First of all, we would like to make you aware of 2 important points. First point, this is a 2-day workshop, actually 2 half days, roughly, a bit more, 1.5 day workshop, showing you absolute basics of an unbelievable broad ecosystem and the things you can do within. Depending on the things you did in the past or things you already tried out, a lot of the things we're going to show you here will be completely new, different from what you've done, what you know. And please, with any other workshop, don't expect to know everything about the things we show you after these 2 days. That's just not possible, not from your side, not from our side. And it's really about knowing what's out there, how it can be done, and why it's useful. Really, one of the basics so that you can navigate this ecosystem and this field better. And in our opinion, if you want to get the most out of it, just show up, which you already did, excuse me, committed, and start using what you see and hear in your own research workflow. Obviously, things take time, but it's definitely worth the effort. In the end, your future self will be endeavorably grateful for you starting to do reproducible neuroimaging. The second part is, it's hopefully, we hope it's your work, so you should take responsibility for the things. So no one can know and be an expert in all these different tools and resources and aspects. That's just not possible as well. But it would be cool, it's actually kind of important and necessary to have a basic understanding of what you're doing and why you're doing it. And a lot of tools out there that let you do a lot of stuff by just clicking buttons. And I think a lot of you know what kind of software I mean, and how most people start doing this type of analysis, which is not encoding, but using one of these graphical interfaces to do some click-to-click analysis. And in our opinion, this is actually very dangerous, or can be very dangerous, because it can prevent a deeper understanding. And the tools presented here, yes, they do require you to think more and maybe in the beginning spend more time on your analysis, but you learn so much more about the data, the things you do with the analysis you want to apply, and are still comparably easy to use. You don't have to have a computer science degree to make use of them. And as a general thing, maybe don't do things just because they're fancy or this is how it always has been done, because both are not very good motivations in science. So for our schedule, we will make for 2 sessions, one today, one tomorrow, each roughly 5 hours length with breaks, obviously, don't worry. And today is going to be basics of reproducible neuroimaging, starting with the introduction, then the derivative introduction, then GB will provide an introduction to reproducibility in neuroimaging. And then we're going to check out how to do version control with the focus on basics and a bit more on analysis scripts, for example. And then talk about virtualization of computing environments and end with a nice presentation in standardization in neuroimaging. Tomorrow, we take it to the next level, talking about advanced concepts in reproducible neuroimaging, starting with a recap of the things we'll do today, then continue our version control adventures, now focusing data and way more. Then also going to talk about metadata and basically end the workshop with starting with a somewhat like workflow one-on-one and connecting the dots between all the different things, how to integrate with one another, to maybe give you some sort of general idea or guideline of what you can do when and where in your research workflow. And then gonna end with a wrap-up and discussion, question and answers from all of you. And if you have time and up for it, we will also have a small social networking event on GatherTown. So for logistics for the workshop, your machine hopefully set up worked. I got a few emails and we had a few people in installation clinics, but so far I didn't hear from anyone else. So I hope that's good to go. You should have a terminal running bash, Git, Conda, and Docker installed in your machine. This is what we're going to need for today and also for tomorrow, at least parts of it. You hopefully should have explored the bash shell a bit in advance. This was one of the course prerequisites we put on the website. And you don't really need to have a thing of Python. If you work with it before, it will be somewhat helpful, but not needed or required. The workshop and the things we do will take up roughly 10 to 15 gigabytes of space, which you can delete at the end of the workshop. If you have troubles along the way running out of memory, just let us know. And obviously, please use the communication channels. So Zoom chat or you raise your virtual hand and ask us a question. As I hope all of you know, we have this entire course in this workshop on a website. I'm just going to post the link again in the Zoom chat, where we basically either already provide or will provide all the materials and things, slides, code, whatever we will do in the next 2 days. Hopefully somewhat easy for you to navigate, revisit later on, and also share with colleagues if you want, of course, because this website will stay up also beyond the workshop for as long as the internet will exist, hopefully. And again, if you have troubles regarding the setup of aspects of it, we have a lengthy section on the setup for the workshop, explaining for each operating system how you get the respective software that is needed for the workshop. And we decided to do that and send you through this somewhat not fun adventure of installing all these things, because we think that if we would provide you with some cloud computing instances, it would basically hinder your adaptation, because if you don't have it on your own machines and are able to use it whenever you want things, then what's that? So that's why we ask you to install all these things so that you're ready to go right after the workshop. Go reproducible. quick thanks before we start to all the instructors that agreed to give a presentation here based on their fantastic experience. So those are the folks you will get to know throughout the workshop, being introduced either by themselves or me when they come up. And obviously also big thanks to the GDPA, specifically Maurizio, who's here. And I think Stefan and Maria are unfortunately not here because of time issues. But I think it's very cool that you have this society with all these people in there that do care about early career researchers and organizing these workshops for you and listening to your wishes and needs and get together and make this happen at a very, very low cost. So this is really cool. If you see the reaction thing on your Zoom, you could use the clapping hand icon emoji now, just to show your appreciation. I don't see it, so I clap and re-clap. Thanks so much for that. And with that, we would be done with the intro.